This video will introduce you to the structure and function of the mitochondrion. The mitochondrion couples the release of chemical potential energy from sugar molecules like glucose or fructose to the synthesis of ATP. Since ATP is the principal energy currency for cells, we need to think about the fact that ATP hydrolysis will be used to power anabolic reactions that normally would not be able to occur on their own due to their positive values of delta G, or free energy. So by coupling the hydrolysis of ATP, a catabolic process, to an anabolic process that is occurring within the cell, we can make the overall chemical reaction spontaneous with a negative delta G value. In terms of structure, the chloroplast, much like the nucleus and the chloroplast, the mitochondrion has an inner and an outer membrane and is the product of endosymbiosis that we've seen earlier. If you were to look at the inner membrane of the mitochondrion, it has many, many folds. These folds are going to increase the surface area available to synthesize ATP within the organelle. So here we can see the folded inner membrane. I'll show you a larger picture on the next slide. And then the mostly smooth outer membrane. And again, the goal of this is to show you that those inner folds increase the surface area available for ATP synthesis. Here's a larger view of the mitochondrion. We can see the outer membrane at its surface. We can see the inner membrane, and then with this cutaway, we can again see those folds that make up the inner membrane of the mitochondrion. Because this mitochondrion evolved from a free-living prokaryotic cell, it has its own DNA as well as its own ribosomes. The ribosomes in the DNA are located within the cytosol of the mitochondrion, known as the mitochondrial matrix. The inner mitochondrial membrane and its folds are known as cristae, and then the space between the outer membrane and the inner membrane of the mitochondrion is known as the intermembrane space. All of these parts are going to be important to us as we continue our investigation of cellular respiration later on in the unit. I'm going to post a link to a short animation/movie that I think you'll find very interesting at this point in the video. I encourage you to watch it. So what is it that the mitochondrion is actually doing? Well, contrary to popular belief, it is not the powerhouse of the cell. What the mitochondrion is responsible for is extracting chemical potential energy from molecules like glucose in the presence of oxygen so as to produce carbon dioxide, water, and energy. But as I've tried to share with you in the past, we can't simply release this energy in little bursts or even in big bursts. Those forms of energy would not be usable by the cell. We can also not simply release the energy in the form of heat because the cell cannot make direct use of thermal energy either. So the goal of cellular respiration will be to couple the catabolism of glucose and oxygen to the anabolism of molecules like carbon dioxide, water, and most importantly, ATP. Just a quick review of the structure of ATP. Remember that ATP stands for adenosine triphosphate. Every ATP molecule has a nitrogenous base, in this case adenine, a sugar, ribose, and then 3 phosphate groups. And to remind you that the chemical potential energy stored in ATP comes from the placement of these individual phosphate groups relative to each other. So that third phosphate group has its own lone pairs of electrons on these oxygen atoms. And by placing that third phosphate group with its lone pairs of electrons closer to, say, the second phosphate group with its own lone pairs of electrons, that electrostatic repulsion between electrons, the repulsion between negative charges, increases the chemical potential energy contained within that molecule, making the molecule high energy. So we've seen in the past that if we had a molecule of ATP, remember 3 phosphate groups, sugar ribose, nitrogenous base adenine, and we were to break off that third phosphate group through hydrolysis, the energy is released. But I'd like you to recall as well that that phosphate group is simply not being released into the surroundings. It's being attached to a molecule, perhaps it's being attached to a protein, perhaps it's being attached to another molecule through the act of phosphorylation, and thus the energy transfer is complete due to phosphate group transfer. So in conclusion, we know that we can synthesize ATP by taking a molecule like ADP and adding a phosphate group. By inputting energy into the system, we can synthesize ATP. Through hydrolysis, we can extract energy from ATP and regenerate the ADP and PI. It will be the role of cellular respiration to produce ATP from ADP and PI by investing chemical potential energy through the catabolism of molecules like glucose.